Hello everyone, welcome back to Chinmoy's Biology channel. So today we are here back with a brand new episode of genetics and evolution. So today's episode we will be stressing on incomplete dominance and codominance. For that we will start with the laws of inheritance and laws of segregation, and we will also do the test cross here. So stay tuned throughout the full episode because many important topics we are going to discuss today. So don't skip in between. We will discuss a huge lot of topics and you will get a good knowledge about that, and your doubts in any topics of today will be cleared easily. So let's get started. So this is where we ended our last episode. We started with this test cross, what Mendel did. That is, he did a cross in F2 generation, that is, he crossed a tall plant from F2 generation with a dwarf plant, right? So in a typical test cross, an organism, pea plants which are taken here showing a dominant phenotype and whose genotype is to be determined. Here we have seen a dominant phenotype. So all are tall and one is dwarf, but we want to know the genotype, right? So it is crossed with a recessive parent instead of self-crossing. So it is a test cross. So tall plant is crossed with a dwarf plant. So it is a test cross. So now here, using the Punnett square, try to find out the nature of the offspring of the test cross. What ratio did you get? Using the genotypes of this cross, can you give a general definition of test cross? So this is where we ended our last episode. So from there, we will continue today with the test cross, and where we will be taking violet colored flower. And this is the dominant one over white color flower, which is the recessive character. So let's get started. So now in the first F1 generation, what we have seen that this homozygous recessive was taken as a parent and a dominant homozygous was taken. So in the F1 generation, we have seen all flowers are purple. Okay, so then in F2 generation, a test cross was done between homozygous recessive and a unknown flower. We can say we don't know the dominant genotype here. We don't know the genotype. So test cross was randomly done between a white flower, that is recessive, white was recessive obviously, and a dominant flower, that is a purple flower. So what we see after this test cross, what was the result? The result was we see half of the flowers are violet. You can see here in the Punnett square also, half of the flowers are violet. So you can see we don't know the genotype yet, and half of the flowers are white. Okay, so they are this unknown flower which was being crossed with, it is heterozygous. And this is here an unknown flower homozygous dominant is there. Okay, so first F1 generation all are purple. In the F2 generation in test cross, we have seen that half of the flowers are purple and half of the flowers are white. Okay, so based on his observation on monohybrid cross, Mendel proposed two general rules to consolidate his understanding of inheritance of monohybrid cross. So what were those laws? These are very important laws. They are the rules which are called the principles or laws of inheritance, right? So the first law, or the law of dominance, and the second law is the law of segregation. So what are these laws? We will understand now. Now the first law, what is it telling? It is telling characters are controlled by discrete unit called factors. So discrete units are there. They are controlled by these units called factors, and factors occurs always in pairs. Okay, factors are occurring in pairs. In a dissimilar pair of factors, one member of a pair dominant, that is dominant, and another pair is recessive. So what are factors here? These factors are only alleles. Okay, these alleles always are paired. Okay, when we are taking a white color recessive, we will take as this is homozygous. When we are taking purple color flower, we will write like this. This is dominant. So one is in capital letters, another one is recessive in small letters. So we know that, we have studied that in the previous episodes. So these are the three important this points in this law of dominance. So the law of dominance is used to explain the expression of only one of the parental characters in monohybrid cross in F1 and the expression of both in F2 generation. It also explains what? That the proportion, that is the phenotype, will be 3:1 obtained at the F2 generation. It is not discussing about the genotype. Okay. Now it is all about the phenotype. Now what is the law of segregation? So in this law is based on the fact that alleles do not show any blending, that both the characters are recovered as such in the F2 generation, though one of these is not seen in F1 stage. Suppose tall and dwarf. In F1 stage, all are tall and no dwarf plant is seen if we are crossing between tall and dwarf plants. But in F2 generation, we are seeing the ratio, phenotypic ratio, that 3 are tall and 1 is dwarf. So there is 3:1 ratio. So there is no blending. So that is few are tall and few are dwarf. So though the parents contain two alleles during gamete formation, okay, so we have studied that during gamete formation 2 alleles are present. The factors or alleles of a pair, they segregate from each other such that a gamete receives only one of the 2 factors. So they are segregating. If we are crossing tall plant with a dwarf plant, so these 2 pairs of alleles in a cross, they are segregating, they are separately written like this. Okay, so we know, we have studied in the Punnett square, so they are segregating. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces 2 kinds of gametes. So if it is, we are crossing between this like this, so these are heterozygous, they will be producing 2 kinds of gametes, each having 1 allele with equal proportion. Is it clear to all of you? So this is what the law of segregation is telling us. Now we will move on to some variation from this laws of inheritance and laws of dominance and laws of segregation. So let us see what is that. Now, in this, we start with the incomplete dominance. So, what is it? When experiments on peas were being repeated using the traits in other plants, it was found that sometimes F1 had a phenotype and that did not resemble another of the 2 parents, but was in between the 2. So, the inheritance of a flower color in dog flower or a snapdragon is a very good example. So during Mendelian's experiment also it was found, but Mendel told some law of dominance and law of segregation, but there was a variation from that. So what is it? So if we are taking this a snapdragon flower, this what is there, it will help us to understand the incomplete dominance. So in a cross between a true breeding red flowers, that is RR, true breeding red flowers, and a true breeding white flowered plants, true breeding white flower plants, in the F1 RR was pink. So the F1 generation RR was pink. Okay. Now what we did, then F1 was self-pollinated and F2 resulted in the following ratio. So this was self-pollinated. Okay, so RR and RR are the parents. Okay, male and female. So what happened in this cross, one is red, 2 are pink here and 1 is white. Okay, so the ratio here we find that 1:2, 2:1. Okay, so that is the genotypic ratio we are finding here. So here the genotypic ratios were exactly as we would expect in any Mendelian monohybrid cross, right? But the phenotypic ratios has changed from 3:1 dominant recessive traits. Okay, what happened was R was not completely dominant here over R, but R should be completely dominant over R. But no, it created a new character, that is pink flower. It was not completely dominant over R, which made it possible to distinguish between RR as a pink flower. But according to Mendel, RR, the dominant character will always be visible. It is a visible character, but it is not happening here. It is showing another color, that is a pink color. Okay, so the phenotypic ratio here is 1:2:1. We can see here, and as well as the genotype ratio is also 1:2:1. So what is the explanation of this concept of dominance? That what exactly is dominance? Why are some alleles dominant and some are recessive? To tackle these questions, we must understand that what the gene is doing. So what is the gene doing? So every gene, as you know by now, contains the information to express a particular trait. So here, what is the trait? Trait is the character, trait is the factor that is either pink or red or white. So to express the trait, a gene is required. So in a diploid organism, there are 2 copies of each gene. We all know that. We have already studied about a pair of alleles. So they are always in pairs. The alleles are always in pairs. So now these 2 alleles, they not always be identical in a heterozygote. One of them may be different due to some changes that is undergone about, we can say that, which you will read in the next chapters as well. Okay, so you will get a modified information about the alleles there. Now, let us take an example of a gene that contains information for producing an enzyme. Okay, now there are 2 copies of gene and the 2 allele forms. Okay, so let us assume that the normal allele produces a normal enzyme that is needed for transformation of substrate S. Theoretically, the modified allele could be responsible for production of normal or less efficient enzyme, non-functional enzyme, or no enzyme at all, right? So this, we have taken an example that our gene is producing an enzyme. So either the modified allele can produce normal enzyme, less efficient enzyme, or non-functional enzyme, or no enzyme at all, because it is modified gene, right? Is it clear to all of you? As it is modified. So what we will see in this case, so in the first case, what happened? The modified allele is equivalent to the unmodified allele. So that is not a modified one. So that is, it will produce the same phenotype or the trait, that is the factor or allele. So result is what? In the transformation of the substrate S. Okay, such equivalent allele pairs are very common. Okay, but if the allele produces non-functional enzyme, that is, if it is producing an enzyme which is not properly functioning, that is, it is a non-functional enzyme, or if no enzyme is produced at all, so what will happen? The phenotype will be affected, and the phenotype trait will only be dependent on the functioning of the unmodified allele. Okay, is it clear? So it is, the phenotype is dependent on the unmodified allele, as in this one no enzyme is produced, non-functional enzyme is produced, so the phenotype may be affected. Okay, so the unmodified allele which represents the original phenotype is the dominant allele. So the unmodified allele which was produced here is the dominant allele, and the modified allele which is called the recessive allele. So we are taking the allele which are producing the enzyme and all. So the unmodified allele here is the dominant allele, and the modified allele is acting as a recessive allele. Okay, is it clear? Now in the example above, the recessive trait is seen due to the non-functional enzyme and because no enzymes are produced in this. Now we will move on to a very important another variation of Mendel's law, that is codominance. So what is here? Let us see. So till now we have discussed about the incomplete dominance. Now here is an important topic that is called codominance. So what is codominance? We are discussing. So we have till now discussed that in F1 they are either resembling 2 parents, like the dominant one or the recessive one, or it was in between that is incomplete dominance. So we have seen in the last slide that 1 red and 1 white is being crossed, and we are getting 1 red, 1 white and 2 pink flowers. So one part is in between 2. But now is a different case. It is a case of codominance. So what is happening in codominance? That the F1 generation resembles both the parents. Suppose a parent, so the character of parent A and the character of parent B both are dominantly present in, like they are partly present. We can say that character of parent A and parent B are partly present in this the daughter or the son, and that is called the codominance. So it is the good example of it is the red blood cells that determine the ABO blood grouping in human beings. Okay. So let us understand what is happening here. In ABO, blood groups are controlled by the gene I. Is it clear? Now, the plasma membrane of red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. Okay, so this gene I is having sugar polymers, okay, that are protruding from its surface, and the kind of sugar molecule is controlling the genes. Okay, now the gene I has three types of allele, IA, IB and this small I. Is it clear? Now the alleles IA and IB, they produce a slightly different form of sugar, while allele I does not produce any sugar. So IA and IB, they are producing a different kind of sugar, and this is not at all producing any sugar. Okay, because humans are diploid organism, each person possess any two of the three gene alleles. Okay, so having two chromosomes of these three alleles, so IA and IB are completely dominant over I. In other words, when IA and I are present, then only IA will represent or express itself, because I is not at all producing any sugar polymer. So it is recessive one. So IA will always be dominant over I. And here the same thing is happening on IB is always dominant over I. So IB expresses. Now, when IA and IB both are present together, they both express. So here comes the codominance. So they are representing their own types of sugars. This is because of codominance. So this is very important. So they are representing their own types of sugar, and this is what is codominance. Now, hence, red blood cells have both A and B types of sugar. Since there are three different alleles, there are 6 different combinations of these 3 alleles that are possible, and therefore a total of 6 different genotypes of human ABO blood group systems are seen. So how many phenotypes are possible here? So how many visible blood group we will see? So table showing the genetic basis of blood groups in human population. So table is showing the genetic basis of blood groups in human population, right? So now we will see what is happening here. So we have seen IA and IB, if it is present, they will codominantly represent. If IA and I is present, it will be IA which is expressing. If IB and I are present, IB will be expressing. Now we will move on to this. So table showing the genetic basis of blood groups in human population. Okay, now allele from parent 1 is IA, allele from parent 2 is IA, both are IA here. So the genotype of the offspring will be IA and the blood group will be A. Is it clear? Now parent 1 IA, parent 2 IB, parent genotype of the offspring IB, blood group AB. Like that only, allele from parent 1 is IA, allele from parent 2 is I. So I, we have told IA will be expressing, so A is the blood group. Like similarly, IB, if it is allele from the parent 1, and IA from the parent 2, both will be expressing. So IA and IB is the genotype and AB is the blood group. Again, IB, if they are expressing, they will be IBB. So B will be the offspring. The blood group of the offspring. Again, if IB from one parent is taken and I gene from the next parent, so we have told that as it is not at all having a sugar polymer, it will not be expressed, and this will be expressed. So IBI, so the B will be the blood group. And if both parents are having I gene, so the genotype of the offspring will be II, and the blood group will be O. Okay, so this is what is codominance. I think it is clear to you all. Now, we will discuss a bit about this multiple alleles which we have spoken in this ABO blood group system. So let us discuss that. Now after discussing about the ABO blood group system, we know that it also provides the example of multiple alleles, right? So here you can see that there are more than 2 alleles, and that is 3 alleles governing the same character, like ABO blood group system. So since in an individual we know only 2 alleles can be present, multiple alleles can be found only when the population studies are made. So when you are doing any population study, multiple alleles can be found only then. Okay, now occasionally we can say that since a gene product may produce more than one effect. So what is more than one effect? We are telling that one gene product, when it is one gene product is there, it can produce more than one effect. So the effect is more than one. So example, how can we take the example? That is starch. During starch synthesis, that is during production of starch in pea seeds, it is controlled by only one gene. It has 2 alleles, B and B. So starch is synthesized effectively by BB homozygotes, and therefore large starch grains are produced. So in contrast, BB homozygous have lesser efficiency in starch synthesis and they produce smaller starch grains. Is it clear to all of you? Now, BB, as it is homozygous, as it is synthesizing more effectively starch, they have a large starch grain produced. But in contrast, the smaller grains like small BB, they are having less efficiency in synthesizing starch. So that one gene is only producing starch synthesizing gene. So after maturation of the seeds, BB seeds are round and BB seeds are wrinkled. So heterozygous, they produce round seeds and B seems to be dominant allele. Okay, so heterozygotes, they are producing round seeds and B seems to be dominant. But the starch grains produced are, we can say that they are of intermediate size in BB seeds. Okay, so the starch grains produced are of intermediate size, so they are neither big neither small. So they are, here we are discussing about the codominance, right? So that will be in between the 2 parents. The character will have the characters of both the small and the big parents. So they are having an intermediate size, and so if the starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. So if the starch grain size is considered as the phenotype, does it mean? That phenotype, when you are externally seeing that, when the external visible character is that one starch grain, one seed is big, one seed is small, and another seed is in intermediate size, so middle size, so neither large nor small. So this is showing the factor of we can say that incomplete dominance. It is not codominance, it is incomplete dominance. Therefore, dominance is not at all an autonomous feature of a gene or a product that it has an information for. Okay, so what does this autonomous term mean? We can say that it is not an autonomous feature. So autonomous means, we can say that autonomous meaning independent feature. Okay, so we have seen, as per Mendel's law, we have seen that we can say that dominance is not an autonomous feature. So it is, according to Mendel, he told that the dominant gene will be expressing in all the generations and the recessive will not be expressed, okay, as a phenotype. But we have seen that there is incomplete dominance, there is codominance, and in codominance we have seen that both the characters of the parents are partially visible in the offspring. So dominance is not an independent feature of a gene or product that has information for it. It is not at all independent. It depends as much on the gene product and the production of a particular phenotype from this product as it does on this particular phenotype. So It is dependent on both the characters of the allele. It is dependent on the production of a particular phenotype, whatever the phenotype is produced. Now we have seen that in case of ABO blood group systems above also, when both the parents are having IA and IB, so both are expressed as AB blood groups in the offspring. So the production of a particular phenotype is like that only. And this product, as it does not depend on a particular phenotype that we choose to examine, it is not as per our choosing. So in that case, more than one phenotype is influenced by the same gene. So we have seen that more than one phenotype, like AB blood group system, it is influenced in other plants also. We have seen that red and white flowers, pink flowers also came into existence. So not only one phenotype or one gene is dominant or dominance only autonomous, it is only independent factor. It's not like that. So all are depending on each other for the production of phenotype, and it is influenced by the same gene. So is it clear? Till now we have made it clear in the monohybrid cross. We have studied about Punnett square. We have studied about multiple alleles. We have studied about incomplete dominance, codominance, dominant, recessive, everything till now it is clear. So in the next class we will study about the inheritance of two genes. So that is the dihybrid cross that Mendel has done after monohybrid, taking the two characters, two traits, that is dominant yellow color seed and recessive green color seed, and round shaped seeds and wrinkle shaped seeds. So this is what we will be doing in the next class. So for that you need to stay tuned, stay subscribed to my channel, and please keep watching my channel, keep watching my videos, and subscribe to it, share it with your friends, and wait for the next video to come. I'll be posting the next video as soon as you go through this video. So stay tuned, stay updated, and thanks a lot for watching my videos, and please do share your comments in the comment box.